Welcome back to AllAboutRx. Today, we're stripping back the complexity of organic chemistry in this explainer. First off, what exactly is it? Well, simply put, it's the study of hydrocarbons and their derivatives. But actually, here's a crucial thing to remember right off the bat. While carbon is the absolute star of the show for organic compounds, there are a few sneaky exceptions. Compounds like carbon monoxide, carbon dioxide, and metal carbonates, yeah, they're actually inorganic. So let's dive into this and make this subject your new best friend.
Here's our roadmap for today. We'll start with the vital force myth, then move on to decoding chemical formulae, inside the carbon atom, rules of electron behavior, and finally, the magic of hybridization.
Section 1. The vital force myth. How one experiment changed chemistry forever.
Okay, so before 1828, chemists were totally convinced that organic compounds could only be produced by living things, plants and animals. We actually used to believe in this thing called a vital force. People thought living cells just contained this mysterious magic necessary to create these molecules. But that all changed thanks to a German chemist named Wöhler. In 1828, he was just boiling ammonium cyanate, which is an inorganic compound, in water. And, totally by accident, he synthesized urea. That's an organic compound that previously had only been obtained from animal urine. With that one boiling flask, the whole vital force theory was literally crushed. To help you lock this landmark historical event into your memory for exams, we've got a high-yield mnemonic cheat code. Just remember, W-U-T. Wöhler, urea, terminated. As in Wöhler's synthesis of urea terminated the vital force theory. Definitely keep that one in your back pocket.
Moving on to Section 2. Decoding chemical formulae. Learning the language of molecules.
So when writing molecules, we use several different types of formulas, and understanding the difference is key. The empirical formula just gives you the absolute simplest reduced ratio of atoms. The molecular formula, on the other hand, tells you the actual exact count of each atom in the molecule. Then you have the condensed formula, which goes a step further by showing groupings of atoms, so you can kind of see how things are arranged sequentially. The displayed formula is the full-blown diagram where every single atom and every single bond is drawn out. And finally, we have the skeletal formula.
Now, the skeletal formula is just a simple zigzag line structure. The rules here are incredibly efficient. Your carbon atoms sit right at the vertices, or the ends of the lines, and the hydrogen atoms attached to those carbons, they're completely implied. You don't even have to draw them out.
And here is why this matters. The skeletal formula is your absolute best friend in exams. Because it completely omits the hydrogens and simplifies the carbon chain into quick lines, it is the fastest to draw and by far the most frequently tested. If you master reading this zigzag, you are going to save yourself so much time.
All right, Section 3. Inside the carbon atom, the star of organic chemistry.
Before we dive into the complex reactions, let's quickly recall the basic anatomy of an atom. Every atom has a superdense nucleus right at the center, containing positively charged protons and uncharged neutrons. This is where almost all the atom's mass is held. Surrounding that is the electron cloud, made up of negatively charged electrons orbiting the nucleus, which actually makes up most of the atom's volume. Now, your mass number is just the total protons plus neutrons. But what happens if that number of neutrons changes? Well, you get isotopes. For example, most carbon is carbon-12, but about 1.1% of it is carbon-13, which just has one extra neutron.
But here's an exam tip you absolutely cannot forget. Carbon's atomic number is 6. It always has 6 protons. While those neutron counts can fluctuate to give us those isotopes, that proton count is the element's exact unchangeable fingerprint. I mean, if you change the protons, it's literally no longer carbon. Section 4. Rules of electron behavior. How electrons fill the void.
So how do these electrons actually behave? Well, they strictly follow 3 rules. First up, the Aufbau principle. This means electrons always fill lower energy orbitals first before moving up, kind of like filling a concert venue from the front row to the back. Second, we have the Pauli exclusion principle. This dictates that each orbital can hold a maximum of only 2 electrons, and they must have opposite spins, so one spinning up, one down. And third is Hund's rule. This basically says every orbital in a subshell gets filled singly with electrons of the same spin direction before any orbital gets a second paired electron. And where do these electrons actually live? They live in orbitals, which have really distinct 3D shapes. The s orbital is perfectly spherical. Then we have the p orbitals, which look like barbells or dumbbells stretched along the x, y, and z axes. And as we get up to the d orbitals, they start looking a little bit like clovers. Now, remembering the order of these orbital shapes, S, P, D, and F, can be a little tricky, so here is another highly effective mnemonic for you. Silly people don't fight. S is spherical, P is your barbell, and then D and F. It's a super simple trick, but honestly, it works every single time to keep those subshells straight.
Which brings us to Section 5, the magic of hybridization, mixing orbitals for bonding.
So what exactly is hybridization? Really, it's simply the process of mixing atomic orbitals of similar energies to form entirely new, equal-energy hybrid orbitals for bonding. This summarizes all that intense math into exactly what you need to know. When one s and one p orbital mix, you get sp hybridization, forming a linear shape with a 180-degree bond angle. If you mix one s and two p's, you get sp2, which is trigonal planar at 120 degrees. And when you mix one s and three p's, you get sp3, otherwise known as tetrahedral hybridization. Take a molecule of methane, for example. This creates a gorgeous 3D symmetrical tetrahedral shape with a very specific bond angle of 109.5 degrees. Now, instead of doing complex orbital math during a test, here is an incredible shortcut. The easiest trick to remember this, just count the electron groups. That means bonds plus lone pairs around the atom. If you count 2 groups, it's sp. If you count 3 groups, it's sp2. And if you count 4 groups, it's sp3. The number of hybrid orbitals formed always perfectly equals the number of atomic orbitals that were mixed. Let's lock this in with the ultimate cheat code for identifying carbon bonds at a glance. If a carbon has only single bonds, it is sp3 hybridized. If it has a double bond, it's sp2. And if you see a triple bond, it is sp hybridized. It really is that fast. You just look at the bonds and you immediately know.
We've covered some absolutely incredible ground today, from the death of the vital force theory to the neat geometry of sp3 orbitals. Master these basics, and the rest of your pharmacy chemistry journey becomes much, much smoother. If you can speak the language of skeletal formulas and understand hybridization, predicting reactions later on becomes a total breeze. Make sure to check the link in the bio for more study notes, and please don't forget to like and subscribe to AllAboutRx.
I want to leave you with one final, provocative thought. Think back to where we started. If Wöhler hadn't been boiling that inorganic ammonium cyanide in 1828, if he hadn't accidentally synthesized urea, how long would the vital force myth have held back the entire field of modern medicine? It's pretty fascinating how one tiny lab accident can completely rewrite the rules of science forever. Keep questioning, keep learning, and I'll see you in the next explainer.